My name is Jeremy Van Cleve, and I'm a assistant professor at the University of Kentucky. And today I'll be talking a bit about some topics pretty close to the kind of research I do, so I'm happy to sort of be excited to be able to introduce some of that stuff today. And I'll try to give you a little bit of a, not totally different, but slightly different perspective on same kinds of topics that folks have talked about so far in terms of evolutionary game theory and thinking about how we can incorporate population structure and the effect of structure on the results from evolutionary games. And so some of it will be sort of focusing quite a bit on that structure component, and then hopefully towards the end we'll actually start to really see how we can measure population structure in kind of a sense that actually comes from population genetics and the kinds of tools that we use in population genetics to measure the sort of size, the extent of population structure. Those kinds of tools will come up in these kinds of analyses. So that's sort of part of the goal, is to be able to see how the same kinds of quantities that one might measure if one were actually to go and gather genetic data, that those quantities also become super important in terms of thinking about social behavior and in the context of structured populations. So to give a context with sort of the same kinds of problem that Sergey was talking about, it's a collective action problem. So we're really thinking about cooperation between multiple kinds of individuals, and those individuals, really that kind of cooperative dilemma, that collective action dilemma, is something that happens even at the smallest scales in terms of or a living thing. So these are dictyostelium, these are these slime mold cells, and they have this interesting behavior I'll mention here in a second. And their kind of cooperative dilemma is of the same kind of has the same qualitative features of the kinds of cooperative dilemmas you think of for much more sophisticated organisms in terms of their development and their behavior, so primates and other animals. So really the main question that a lot of these, that I'll be sort of thinking of in the back of my head as I develop a lot of these tools, is how do these kinds of cooperative behaviors evolve? And just to sort of give sort of a very simple definition of what I mean by cooperation, I sort of use that generically to mean any kind of behavior that implies a cost to a focal individual when it's improving the state of its partner. And then the partner, of course, then receives some kind of benefit, and we'll sort of often talk about payoff as a way to measure that benefit, but ultimately that benefit has to accrue in terms of increased fitness, so increased survival and reproduction for the partner. And so then that's just sort of said, this improved state has to improve fitness, and that's really what we're measuring in terms of the evolutionary quantity in the long run. So even the simplest organisms have this kind of collective action behavior. So this is dictyostelium, and again, so it has this interesting behavior where when it's under nutrient stress, it follows a certain sort of set of chemical gradients, and individual cells stream together to form kind of a collective whole. That collective whole, this sort of slug, it migrates on the environment, and at some point it forms this stalk-shaped structure with a spore at the top. And the individuals in the stalk are individuals who will end up sort of forming a structural element, and they end up dying. The individuals in the spore get to survive. They hopefully disperse off to some environment which ideally has a better food situation, resource situation than the one they're currently in. So that's certainly a very cooperative activity, given that the ones in the stalk are essentially sacrificing themselves for the ones in the spore. And so this sort of gives us an obvious sort of cost and benefit breakdown in terms of the who's paying a cost, what that cost is, and who's paying a benefit, and what the benefit they're getting from that. So really, even though we know these kinds of cooperative interactions happen even at the smallest scales, we sort of like to be able to answer a question about how do those cooperative interactions work together at small scales to build up cooperative interactions at larger scales. So we could think of genes sort of coming together in a sense you could call almost cooperating to form chromosomes, that at a higher scale prokaryotes in the form of sort of ancestral unicellular organisms that were the ancestors of mitochondria and chloroplasts, they came together to form eukaryotes. And going up, we can even think of clonal cells coming together to do sexual reproduction, independent living cells coming together to form multicellular organisms, and then finally, these independent individuals coming together to form social groups, and those social groups becoming so social that sometimes they can be thought of as an evolutionary individual in their own right. So this sort of way of thinking about it as a hierarchy was sort of first talked about by John Maynard Smith and Urs Shattenry, and they call it sort of these transitions at each level. They sort of label the transition in individuality with the idea that once that sort of cooperation became strong enough between individuals within a group, then that group itself could be an evolutionary individual in its own right if the interests of individuals in the group were not tightly aligned enough. So we'll keep an eye on this in some sense, that we'll be able to start to attack the tools that we'll use to look at population structure, and multi-level selection will allow us to start to think about these transitions more quantitatively. So when we think about cooperation happening at these different levels, it really happens in a sense on two different scales. It happens on a scale sort of within groups, and it happens on a scale between groups. And so typically when we think about these different scales, when we think about within groups, the kinds of mechanisms that kind of can allow individuals to cooperate are the fact that they can have plastic behaviors. So, and then when we think about between groups, the kinds of mechanisms that allow individuals to cooperate are often have to do with population structure and have to do with whether individuals are interacting more often than not with individuals who are related to them, with kin, or whether they tend to be interacting with in-groups, and those groups tend to be competing, as Sergey was talking about, competing with other groups. Yeah, so everything I'll be talking about today will be intraspecific cooperation. A lot of the, you can definitely export some of these ideas of thinking about mutualisms between species. Some of this stuff about thinking about the effects of kin, there aren't really kin between species, but you can still get correlations, even genotypic correlations between, so you could have a mutualist a particular genotype of mutualist that's really good, for example, rhizobia, which provides nitrogen to plants, could have there could be really helpful genotypes of rhizobia, and those genotypes might interact preferentially with certain genotypes of plants that provide them with a high amount of carbon to feed them. So some of these ideas definitely translate across. It becomes a little bit more tricky, though, because you can't have that. They're not reproducing together like you have within species. So when we think about these plastic behaviors, the tools that we tend to use are the tools of game theory. So we're looking for Nash equilibria. We're looking for evolutionary stable strategies. And then as well, we're looking at one thing we'll start to talk about is that enables this kind of cooperative behavior to really persist within groups is the ability of individuals to interact repeatedly to be able to play the game over and over again. So we're thinking about things like examples like tit for tat, if you've heard of that kind of repeated game strategy, the ability of individuals to punish one another if they've seen individuals not cooperate in previous plays of the game, etc. And then at that group level structure, it's really this multi-level selection effect, this fact that we can have sort of effects within groups and then effects between groups. And that, while in itself actually creates selection for cooperation, we can combine these two things. We'll see how we can combine the effect of cooperation within groups due to the fact they might be repeatedly interacting with one another. And we can see how that interacts with the effect of population structure to produce sort of an emergent level of cooperation that's a combined feature of both of those elements. So this is sort of the broad scale outline. So I'll talk a little bit, I'll start off by presenting a model of responsiveness, this sort of within group cooperation example of a repeated interaction of playing a game over and over. And then I'll go on and talk about the price equation, which is a way that we'll sort of use to, at least initially conceptually, structure how the selection can affect individuals, whether they're kin or not kin, or individuals within groups or between groups. And then I'll use the price equation then to be able to show how we can have this effect of multi-level selection that also incorporates the ability of individuals to be playing games within their group, to be doing tit for tat or being reciprocators or non-reciprocators. And then the second half, I'll get into a little bit more specific detail about how we can start to make the model more dynamic. So this previous part will be a little bit less dynamic in the sense that we won't really be kind of tracking gene frequency changes over time, but in the second part we'll be really starting to provide much more of a modeling framework to show how one can do that, although it will still leverage some of the work that we talk about in the price equation. And we'll be able to see then in a more specific example of population structure, how structure really starts to affect the conditions for the evolution of cooperation. So getting rolling on this first part for within a population, thinking about how we would start to model cooperation emerging. So we can imagine these two capuchin monkeys. They're potentially playing a cooperative game, or there's a potential cooperative interaction. And so they may have two options to either cooperate or defect. If they cooperate, maybe it's a food sharing interaction, they're sharing food with their partner, then there's going to be some kind of benefit to the partner for receiving the food and a cost to the individual providing it. And if that benefits and cost are positive, which we just typically assume for these kinds of behaviors, then there's a good outcome if they both cooperate. But because of that unilateral cost of investment, then we end up with this kind of prisoner's dilemma game where individuals will, at least if they're only playing the game once, won't have any incentive to cooperate. So really, it's strategies in this repeated game. If we allow them to play over and over again, then we can see how they might respond to the actions that their partner played in the last round. So if their partner cooperated last time, different strategies will react to that past cooperation or defection differently. So this is sort of an example of some common strategies that people have studied in the evolutionary game theory literature. So one could sort of decide to always play the same strategy over and over again. One could start off playing a cooperate strategy, but then defect forevermore if their partner decides not to cooperate and their partner defects, and so on. So one sort of long sort of running theme of research in evolutionary game theory has been to look at the which ones are sort of more evolutionarily productive, which of these strategies against which other strategies. And they've started up with some famous tournaments where people sort of set up computer tournaments to play these different strategies against one another to see which ones persisted over time. So usually the idea for these repeated game strategies, when we think about it from an evolutionary perspective, is that every time they play the game and they receive sort of a benefit minus cost, that benefit is being that benefit minus cost is accruing to their fitness in terms of their chance of surviving or the number of offspring they have. So though each time they play the game, there's a possibility that benefit and cost, the longer they play the game, that those benefits and costs are discounted over time. And potentially those discounts can be due to the fact that interaction might be broken off with some probability. So you can imagine that you sort of, the first time you come together to play the game, you play it once, and then with the probability omega, you continue the interaction. And so over time, you can imagine that the length of the interaction, the probability it reaches n stages is going to get smaller and smaller as n grows larger. So you can think of this omega then in some ways as a discount, that the larger that omega is, the less that you're discounting those interactions, because that's the higher chance that you'll actually end up cooperating with that partner many more times in the future. So then if you want to sort of add up the total number, the amount of payoff for this grim trigger individual with this suspicious tit for tat strategy when they're playing each other, then you would just add up the payoffs for cooperate and defect here, and then add the payoffs for cooperate and defect here times omega squared, and so on. So one thing that's really important about these repeated interactions is, for example, in this grim trigger strategy, that in this first interaction, they start off with a cooperate, and the rule then for grim trigger is that if their partner defects at any point in time, they respond by defecting and they continue to defect. So simply by the fact that they play an individual who starts off by defecting, they gain some knowledge about the strategy their partner is playing. And as they continue to play the game longer and longer, so these games have very sort of simple rules about how individuals respond to their partner's last plays. But as many of those last plays accumulate over time, you get more and more information about the kind of type your partner has. So over these really long interactions, you can sort of figure out whether your partner is playing effectively figure out whether your partner is playing as a tit-for-tat strategy or a grim trigger strategy. And that's that accumulation of information that allows you to sort of escape, in some sense, the prisoner's dilemma and allow you to actually have cooperation emerging from some of these repeated games, even though it wouldn't have emerged from just a one-shot game. So it's really this sort of notion that these repeated strategies, they vary in a certain sense roughly along two axes. So one can think of them varying along an axis where some of these repeated strategies are more likely to lead to cooperation, and other ones are more likely to lead to defection. And then some of them might be more responsive to the actions of their partner, and others might be less responsive. So to give an example, this is a tit-for-tat individual who, if they play the strategy their partner played in the last round, and this is a tit-for-two-tats individual who will only retaliate by defecting if their partner defects twice in a row. So if the tit-for-tat individual plays someone who always defects, then immediately after the partner defects, they continue defecting. And if this two tits for tat plays against an all defector, it takes two of these defections before this individual starts defecting. So in a sense, this two tit for tatter is a little bit less responsive to the immediate change in its partner's action. So even though this tit for tatter and these two tit for tatters might have the same level of cooperation in a sense that they both sort of end up with when they play each other, one is a little bit less responsive to the actions of its partner than another. So this is sort of what I want to get at a little bit, is this level of responsiveness to sort of see how this affects the outcome of the cooperative game. So we can think of this as sort of this has come up a couple different times in the literature before as they've called it in repeated games direct reciprocity, sort of a you scratch my back, I scratch yours kind of interaction. And then this sort of well-known biologist, Bob Trivers, wrote about it in the 70s, and he called it reciprocal altruism. So very similar ideas, and game theorists have been looking at this kind of notion of individuals responding to the past plays of their partners very commonly. So we'll just look at a very sort of relatively simple model of that can give us some intuition about how this might work. So what we're going to do is we're going to set up an index for this responsiveness, and we'll just call it rho, to see on its effect of cooperation in a single population, and we can then later on take that and put it into this context of population structure. So the model will start off, and we'll sort of assume there's two different types of individuals. There's an individual who's a sort of an intrinsic cooperator type who likes to play cooperate and plays cooperate with probability 1 minus rho. And with probability rho, that individual reciprocates its partner's last action. Then likewise, there's going to be a D-type individual who's an intrinsic defector who, with probability 1 minus rho, defects. And then with probability rho, reciprocates the last action of its partner. So if we play these two types together, if we play two C-types together, since the C-type intrinsically wants to cooperate with probability rho, if two C-types are playing and they're intrinsically cooperating, they always cooperate. And if those two C-types are reciprocating with probability rho to what their partner did, they're also cooperating, so they're always going to cooperate. And then likewise, two D-types will always defect when playing each other. So the only part of the interaction we have to look at is what happens when a C-type plays a D-type. So we'll write out a matrix for this when this happens. So we can sort of imagine that there's 4 different possible states for individuals to be in this kind of interaction. So where the C-type is cooperating, the D-type is cooperating, C-type is cooperating, D-type is defecting, and then so on. And so this matrix will then show us sort of the probability that you move from one of these states, from, for example, CC, to probability to DC or DD, and so on. So you just sort of populate it based on this interaction that they reciprocate with probability rho and do their intrinsic preference to cooperate or defect with probability 1 minus rho. So with that matrix then, to figure out what happens in the long run, we can just look at the stationary distribution of it. So find its left eigenvector, and that's what this happens to be here. So you can so this is that cooperates with probability rho, or proportional to probability rho, individuals are cooperating. So they have a higher chance than of doing this cooperate defect, where this is the C type and this is the D type, and then rho squared in rho. And so, well, to figure out what then the costs and the benefits are in terms of payoffs for these two different types, for C intrinsic for the C type and for the D type, we can just look at the payoffs for each of those different outcomes. So if individuals are both cooperating, they're getting B minus C. If there's a cooperation and defection here, it's minus C for that individual is cooperating, B for the defector when their partner cooperates, and then 0. And so what we'll assume for this, just for simplicity for this case, is there's no discounting. So that's sort of a mega probability we'll assume basically to be very close to 1. So individuals are always they're reciprocating the interaction long enough so that they get to this equilibrium distribution of the Markov chain, so that we can just simply calculate the payoff by multiplying the payoffs times these probabilities. And then what we'll assume then is that those payoffs, that's going to be equal to their fitness, the equilibrium actions in the game. So if we, to sort of set up, if we want to calculate the fitness of this C-type individual, this intrinsic cooperator, we could assume that there's a frequency P of those C-types in the population. So if there's basically P of them, and this individual's we want to know the fitness of a cooperator, then with probability P, he's going to run into another cooperator. The payoff for that is B minus C. And then with probability 1 minus P, he runs into a defector. And then we just have to look to here to figure out what happens with this cooperator plays a defector. And we just sum up the benefits that cooperator would get across the probabilities of these 4 different outcomes, which is just this quantity here. So we simplify that a bit, and we get something that looks relatively simple here. We do the same thing for the fitness of a defector, given the frequency of C types is P. And we get a simple expression here. And then if we just want to know when is a cooperator going to have more fitness than a defector, we just say, you set that up as an inequality. And a lot of stuff, these two terms here cancel, and then leaving us just with B rho minus C greater than 0. So this is sort of familiar from a little bit before maybe, or if you've seen Hamilton's rule and you've ever sort of read anything about kin selection. So this looks a little bit like Hamilton's rule. And so it just says that as long as the benefits times the level of responsiveness is greater than the costs, then these C types will increase in frequency. And so this result, although we kind of set up the game in a very particular way, and we gave them, we set up the structure of the game in a specific setup, this kind of result has come up a couple different times as an ESS result where people have looked at this continuous iterated prisoner's dilemma. So this idea, this benefits the game being repeated over many times is the iterated part, and continuous just that their investments into the game, into how much they're cooperating and defecting is a continuous value. And here that continuity is really being provided by how much they're reciprocating with their partner. So the next step is to sort of build a little more of a framework to think about how this evolution can happen in structured populations. And so for this, we'll talk quite a bit about the price equation. So before we talk about the price equation, we sort of talk about, I'll give a little bit of a historical context of why we'll talk about it. So if you're a population, oh, yep.
So on the last model, why would rho be the same between cooperators and defectors? Wouldn't rho be a parameter that itself evolves?
So in this setup, this is sort of a very simplified one where we've set the rho to be the same for the cooperator and defector types, and that's just what this thing simplifies to here. It's a little bit more complicated if they're different. If you let rho itself evolve, what happens is actually this thing, if this becomes the ESS condition. This thing is set equal to 0, and you have a continuum of neutrally stable outcomes, where as long as you set rho, you can find a benefit and cost that's a neutrally stable ESS. So that's it's actually sort of a weird feature of these iterated games, that at that outcome you can find sort of the efficient, so when rho is equal to 1, individuals are getting the benefit minus the cost, the maximum payoff they could get. And when that rho is equal to 0, they're getting minus cost, which is the least they could get. and any of those outcomes is neutrally stable in that kind of setup when you let rho evolve. So if you've ever looked into game theory a little bit, then there's a theorem from game theory called the Folk theorem, which says that in these repeated games you can get any of those repeated games or any possible payoff outcome for the partners in a repeated game is a Nash equilibrium. And that's sort of the same idea. There's a kind of neutral stability among these different equilibria. So it's often the case that we need to add in things like population structure to look to see when we get these, when we get a specific value of rho evolves over another. That was probably a lot for your question. Does that help at all, or?
Kind of. I guess I'll look at that thing.
Yeah. So it's hard to give intuition for why when you let rho directly evolve that you can get sort of many values of it evolving, but it's sort of it's almost a pathology in some sense of these kinds of repeated game models.
I guess on the other side, if the NCR represents that at least for a given action or interaction, then rho is constrained.
Correct, yeah. So if you fix those things, then rho is going to be constrained. That's right. So yeah, once you start to put some constraints on it, then that indeterminacy would evaporate. But the intuition is to want to allow that responsiveness to evolve directly, and it turns out to be the sort of more difficult way to attack the problem. Okay, so going back to the sort of a motivation for why we'll end up talking about the price equation is if you're a population geneticist, then often what you're used to doing is looking at these dynamical systems where you track the frequency of a gene at one locus over time, and then maybe you want to track the frequency of another gene, and you have to track them together, and so you build a very complex set of recursions. So this might be what you would have to do. You'd have these two genes, A and B, maybe with two different alleles, A1 and A2, and B1 and B2. So if you were to track the frequency of these genes over time, you'd have to create a dynamical system that had 4 equations. And then, so that might be something that you can do with some amount of ease. And you might even be able to get some nice analytical results, and people have done that. But now you're interested in population structure. So even if you had just 2 populations, you're going to increase pretty dramatically the number of state variables you have. So you're going to have to track the frequency of these A1, A1, B1, A2, B1, and so on in population 1. And then you have to do the same thing for population 2, leaving you with these 8 recursions. And then you can imagine as you scale up the number of different populations that are possible, the number of recursions grows really, really large, and it becomes quite difficult to analyze. So although it's possible even in these cases to do some nice tricks and to be able to look at, for example, is there maybe an equilibrium point where this A1 is stable to invasion against A2, or is there maybe a point where this A1, B1 is stable to invasion by any other alternative? It still requires doing this analysis on a very large matrix and finding the eigenvalues of that large matrix. And again, that's sort of can be a very complicated process. So these kinds of, and then if you wanted to add these, that's just sort of talking about the case when it's deterministic, and if you want to add stochasticity to that kind of system, it makes it even more difficult to analyze. So one alternative that people have looked at for looking at these systems, in some sense, borrows a little bit from what you could think of are like how physics looks at the interactions between many particle systems. And so where instead of looking at the frequency of a certain kind of particle, you actually have initially a state variable that tells you the status of that particle. So like we had heard a little bit about these in the network models examples, we have a state variable that measures whether that individual has the behavior or doesn't have the behavior, whether they're activated or not activated. So initially, these kinds of models are more complex because you have a state variable for every single individual in every single population. But they tend to simplify pretty rapidly because what you can end up doing is calculating the average frequency across the whole population, across all sets of subpopulations. And that average frequency becomes a function of other statistical quantities. So it becomes a function of the variance in that frequency across all of those populations, and then maybe a third statistical moment and a fourth statistical moment. So you'll get sort of an equation that tells you what the mean change in that allele frequency is. Maybe that's the frequency of this cooperator allele, or it's a frequency of something else. And it's going to be some kind of function of the mean frequency in the initial time step, the variance in the frequency of the initial time step, and so on. And then what you do is, if you only care about, say, the change in the mean, maybe because you want to know, does the mean value of this variable increase? Does the mean fraction of cooperators in the population increase? Then one thing you can do is you can just sort of assume that this variance kind of takes a fixed value. And if you do that, then you know what this variance itself is not changing over time, and you can continue to stick it into this equation for the change in the mean. And so what this kind of method allows you to do is you can study really complicated stochastic processes like this by looking at just the mean, or if you're interested in looking at the variance, looking at that as well, and then sort of cutting everything, not concerning yourself with any other of the higher moments of the distribution. So this kind of approach in general is called a moment closure approach. And so what that means is sort of each of these, you're closing the moments, you're sort of deciding that after the third moment, that's the only one you're going to care about. You're going to allow, you're going to track the dynamics of that third moment, and the fourth one you're going to forget about, and so on. And so what we'll see that there's, later on, that there's a way to sort of do that moment closure in sort of a controlled way from a population genetic point of view. So an example of this kind of statistical approach comes from quantitative genetics, which actually sort of is inherently based on this way of tracking the change in gene frequencies or tracking the change in phenotype frequencies in populations. So if you've seen something like the Breeder's equation in population genetics, which tells you the change in, say, body size, a trait over time, and it tells you that it's the strength of selection times this heritability, which is really just a measure of the variance in gene frequency in the population. So this is just saying the change in the mean is a function of the change of the variance in the population. So often when quantitative geneticists will do models, they'll just assume that variance is a constant and allows them to make a prediction about what the change in the mean is going to do from time step to time step. So really often what they do that just sort of saying again, that's how they get this moment closure property, is by assuming that those genetic variances are constant. So in some sense, the general approach for how to do this was kind of introduced into evolutionary biology by originally an engineer named George Price. And as well, it sort of was parts of it were discovered independently by quantitative geneticists, such as Alan Robertson. And what the Price equation is just a general way to express this change in either allele frequency or phenotype frequency. And we'll dissect that into its component parts in a bit. And it's often associated, if you've done any reading in the evolutionary biology literature, it's often associated with analyses of group selection or multi-level selection, but it's quite general that way, and you can use it whether you're sort of interested in those problems or not. And so it's particularly useful for population structure because you don't necessarily have to assume anything about the population structure initially when you start to analyze the population you're interested in with the Price equation. And then you can allow that sort of can help you figure out how that structure affects this change in gene frequency over time. It tends to be, for whatever reason, an equation that's both sort of oversold by some people who are huge fans of it, as well as highly criticized by others. And so I won't talk about either of those features of it today. But just to say that it's I do think it's somewhat useful for looking at structure, and it'll come up in a couple different places, even in the second half when we really start talking about fixation probabilities and coalescence times and more population genetic ways to attack the problem. So to start just describing what it is, we'll assume that this we're going to look for each individual. We'll let this Zi be the value of some phenotype in individual I. So it could be an allele frequency, it could be the body size of that individual, it could be the amount of investment that individual puts into cooperation. And then the change in we'll let Z be the mean value of Zi averaged over all individuals I. So the change in the mean over a single generation is delta z. And you can sort of write it out kind of elementary by just counting up what it was in the current time step. So this is the frequency of individuals. So this is zi is the individuals of type I essentially who have a value for z of zi. So that could be sort of individuals. There's a class of individuals with a body size, a certain body size. That's given by this. And then that's the frequency of those individuals in the population. So the change then is just those same individuals in the next time step, what their body size is. That body size, it could be the same. It could have changed because of a mutation. And then the frequency of those types in the next generation. You just average those up and subtract them from what they were initially. So that's just sort of one way. That's just writing down just by knowledge of how population changes over time what that delta z should be. So this frequency in the next generation, if we assume it's changing because of some effect on fitness, then those individuals of type I will have a fitness wi, and then their relative fraction in the next generation is just their fitness times their frequency divided by the mean fitness in the population. And so if we set then this delta for the change that each individual goes, their type, so if remember there was a component, their body size could be changing or because of a mutation, that would be captured in this delta z. Then we just sort of insert that into this z prime into here, and then this p prime into here, ending up with this expression. We just rearrange it, pull this term in front here, this w bar here, and this delta z here. And if you just read off what these two expressions are, this first one you can write as effectively the covariance between the fitness of individual I and its phenotype, plus this expected change in the weighted by fitness, this expected change in the phenotype of individual I. And so this itself, this is just the price equation. Yep. Right.
With the Breeder's equation, I can estimate the selective advantage, I can estimate heritability, I can make a prediction about the response to selection. It seems to me you can't do that with the price equation. The price equation is a definition of genetic change, not a prediction of genetic change.
Well, you can immediately get the Breeder's equation out of the price equation. We'll get a couple of results from that here. We'll get the Fisher's fundamental theorem out of the price equation in just a second. But the Breeder's equation, the reason it can make a prediction is because it makes additional assumptions. It makes an additional assumption about the fact that this genetic variance in the population is constant. So if you don't make that assumption, then it has just as much predictive power as the price equation does originally, which is that it just tells you what happens over one time step. So these two components really, one reason that people are interested in sort of thinking about and using the price equation is you can think of these two components as sort of partitioning the evolutionary change into two different pieces. The first component, the covariance, is the evolutionary change in this allele frequency or change in this phenotype due to natural selection. So because it's really measuring the statistical association between fitness and that trait. And then the second component, this expected change, is this evolutionary change due to the sort of imperfect transmission from that trait Z in the current generation to whatever it is in the next generation. So that imperfect transmission could be because Z is mutating. The body size, the genes that underlie body size have a mutation in them going from the current time step to the next one. So this sort of would capture any of those changes in transmission that aren't due to natural selection. So mutation, non-random mating, segregation, distortion, migration, all these different effects would be sort of thrown into that big bucket of this second term in the price equation. Whereas this first one is really just the effect due to natural selection. So to give you sort of an example, this is how you can sort of get some normal population genetic results out of this. If we allow this Z to be the frequency of an allele in a population, And so for each individual, it's either 0 or 1. We're going to assume individuals are haploid. They only have one copy of every gene. And then we'll say that individuals who have the gene have a fitness of 1 plus S. So that's when pi is 1. And individuals who don't have the gene have a fitness of 1. So we can figure out then then we'll let these genes mutate as well. So with probability 1 minus mu, then the next, it's transmitted perfectly to offspring, and it's the same value. And with probability mu, it changes to the alternative type. So to maybe a little, from a big A to a little a or something like that. So then this is the total change in that, just due to the mutational effect. And so then we can, just with these components alone, we can calculate these two different pieces, the covariance, the change due to selection, and the change due to transmission. So if we just plug those two pieces in, the answer we get is this. So this change due just to selection is just this selection coefficient S times the variance in allele frequency. So this is basically very similar to the Breeder's equation. And this expected change here is this expression here, this mutation rate times this component here. So then the whole change in the allele frequency then is just the sum of these two pieces, which is just this equation. And to show you that it gets out something that you're probably used to seeing, if you set this equal to 0 to look at when you get a balance between mutation and selection. So here, this individuals who have the allele have a benefit, but there's some mutation away from that beneficial allele to the deleterious allele. So you set equal to 0, assume that mutation rates are small, and then if this is the detriment of the deleterious allele, then it's basically 1 minus the mutation rate over that selection coefficient. That's a classic result from population genetics. So what we'll look at, right now are sort of 3 conceptual applications of the price equation, 3 different ways that we can use it to sort of think about how fitness affects, how selection changes the frequency of genes or phenotypes in populations. And so those 3 fundamental things are something called Fisher's fundamental theorem of natural selection. We'll see how we can get that from the price equation. We'll extract Hamilton's rule from the price equation, this effect of cooperation when you're interacting with some individuals who are relatives and some who are not. And then we'll look at how we can use it to understand multi-level selection. So starting with Fisher's fundamental theorem, this was written by Fisher in a book in 1930, and it wasn't really a theorem because he didn't really prove it or say anything about it that was too understandable at the time. It's been something that people have spent a lot of time arguing about, but he said this thing just in text, that the rate of increase in fitness of any organism at any time is equal to its genetic variance in fitness at that time. So this was sort of a nice way of encapsulating how sort of our Darwinian, if you want to call it that, notion of how natural selection could change, could sort of increase the fitness of organisms, how they could become better and better adapted to their environment. But in part, it became difficult because if you treated this as something that happened always, it would suggest fitness was always increasing in populations. And if you've done any kind of evolutionary model or something, you know that that's not entirely true. And so we'll be able to see how this actually starts to boil out. So one thing that this does have is if it implies that if there's no genetic variance in the population, then the population is there's no change in that allele frequency, there's no change in that phenotype, so that's kind of an equilibrium condition. And as well, if it's positive, then it implies the fitness is always increasing. So this is sort of a stability condition as well. So you get both of those from the fundamental theorem. And in one way, it's sort of you can sort of think of it as like a mathematical way of, again, talking about this idea of Darwin, and it's similar in some sense to sort of these kinds of welfare theorems from economics that sort of mathematize, in a sense, that idea of the invisible hand, increasing the outcome in terms of making an economic outcome more efficient for everyone who's involved. For example, if they're in a market trading with one another. So one way to start doing this is we'll write down this phenotype of an individual I, and what Fisher was a statistician, and so Fisher and developed a lot of statistics related to regression and analysis of variance and so lots of things. If you're using any kind of quantitative method, you've probably used something that Fisher touched. So what he was interested in is using regression, and in part because regression allows us to pick apart this phenotype in terms of additive, linear regression at least, in terms of additive effects of genes on a phenotype. So that's all we're doing here is just taking the whenever these genes are for each gene J, and has some kind of additive in its value. Sorry, so this is for, right, so this is for each gene J, its value in individual I, and then has that each gene J has a value, some additive effect Bj on this phenotype. So then you add this whole thing up together and you get basically a single additive effect of genes on the phenotype, and that's called, in quantitative genetics, a breeding value. So one nice thing about this is that if in this regression, these are the residuals from the regression, that these residuals are uncorrelated with these additive effects. So it's sort of something we have to keep track of when we'll do the proof here in a second. And the residuals also have a mean of 0. And these are properties just of applying a least squares regression. Doesn't make any assumption about the distribution of any of these variables. So we'll plug this equation for the phenotype for individual I into the price equation. And so since this thing has mean 0, then we can get rid of this. So this all the change in the phenotype it's going to be a change in its breeding value, and these additive effects of genes on phenotype G. So then that's really just this price equation in terms of these G's instead of the Z's. And for Fisher, when he was thinking about the fundamental theorem, he talked about that it was the rate of increase of fitness. So his phenotype he was interested in was fitness. So we're going to plug in, instead of Z, we're going to plug in W. So we're going to assume that fitness there's some additive effect of genes on fitness. And then all we do is once we plug in fitness into this component here, this has a mean 0, so it goes away, and we just get the variance in fitness. The variance in the genetic variance plus this transmission term. So when Fisher wrote his book, he was very kind of unclear he wrote in sort of opaque terms about the deterioration of the environment. And people who have read the book since then if they give him a charitable interpretation, they interpret that to mean that this change in transmission, so the effects of the environment causing mutations on genes, that those effects he was excluding, he was ignoring them. So for that purpose, we'll set this transmission term to 0. And so if we do that, then all we're left with is this variance in gene frequency, and that's exactly the fundamental theorem. So in the next step, we'll use the same thing, the price equation, and we'll get, go ahead.
Sorry, the GIs, so go back, what exactly are the defined?
So the GIs are the linear regression, the linear component of the effect of genes. So there could be many genes, J, having an effect on this phenotype. And if you regress the effect of those genes on phenotype, and these are the regression coefficients Bj, and you just sum up across all those genes, J, times their regression coefficient, and that's this GI. It can be across genotypes. It's a sort of as general in quantitative genetics. You can expand this definition of breeding value to be multi-locus across individuals who are haploid or diploid or polyploid. Absolutely.
How can you handle, so if I start with a population of heterozygotes in a situation where heterozygotes have an advantage, so I have a fitness decrease after one generation, where does that show up?
So that shows up in this deterioration. So again, that's sort of I guess the charitable interpretation is to say that when Fisher talked about the fundamental theorem, he was excluding this term, and that term captures any of these additional effects. So this is just the sort of effect of selection due to additive effects of genes, and anything that's not an effective selection due to additive effects is captured in the expectation term. But we know it has to be captured in one of these because we've written the change out exactly. So.
And so what was Fisher's reason for assuming that expectation to be 0?
I don't know if there's any agreed upon conclusion about that. It probably depends on who you talk to. I mean, I guess I would say because he cared about selection. And he was interested in understanding selection, and he didn't care about anything else, really. And so there was sort of a larger historical, historically there was a larger argument between Fisher and Sewell Wright about whether selection was super important or how much population structure had an effect on creating complex adaptations, and Fisher tended to care much more about selection. So maybe that's one potential reason, but I don't really know. I'm sure could be the subject of many future papers on the history of science. But so the next thing we'll look at is in a structured population, we'll look at deriving this Hamilton's rule formula, which we'll show in a second. So the effect, how individuals might have social effects on their partners, depending on whether their partners are genetic relatives or not. So when Bill Hamilton wrote his paper, this is where he introduced his eponymous rule. He said a gene may receive positive selection even though it disadvantages its bearers if it causes them to confer sufficiently large advantages on relatives. So the rule then is this. So the rule is this, whatever this fitness cost is, if it's outweighed by that benefit from helping, times the relatedness between the individual who receives the help and the helper, so this genetic relatedness. If this whole thing is positive, then that helping behavior can increase in frequency in the population. So really, he was capturing just sort of a relatively intuitive notion that the effect of natural selection on a gene is not just on the copy. When you think of a focal individual who has a certain number of offspring it produces, the effect of selection on the gene isn't just on the copy of that gene in the focal individual, it's on the copy of that gene in every individual in the population. So in a sense, all Hamilton's rule is really doing is sort of providing a certain way to formalize that, to write that down into an equation. So this thing here, this minus C plus RB, it's something that takes a name. Sometimes you'll see in the literature it's called the inclusive fitness effect. And sort of one thing to keep in mind about it is it's not, when you're sort of measuring something like this, you're not actually measuring some special notion of fitness necessarily. You're still doing what you would normally think of doing by counting up all the number of offspring produced by all the individuals in the population. It's just that we're by using the price equation, we're partitioning that counting effort into a certain way where we can extract from it this effect of genetic relatedness. So then the first step, it's going to be like before we did for Fisher's theorem. We're going to write a regression for the phenotype of an individual. And sort of this is, again, similar to sort of how lots of people do this in quantitative genetics when they're looking at even morphology changes, brain size or body size, all kinds of features. So it's not social evolution and people interested in cooperation are not the only ones who do this in terms of writing regressions. So this fitness of individual I is going to be then the phenotype of it is going to be a function of its phenotype times a regression coefficient, which is the cost, plus the mean group phenotype times a regression coefficient, which is a benefit. So if we're then thinking about a cooperative activity or like some kind of collective action, then you'll receive the benefit from when your partners or your group members are helping you, and then you pay a cost if you're contributing to that help. And so then we can just, because each of these components are themselves a regression of that phenotype on some genetic component, we can just write that out and collect everything that was a residual into the residuals here. And so this is just the genotype of individual I, the cost, and then the mean genotype in its group times the benefit. So then all we have to do is plug this into the price equation, into here, plug the fitness into here, and then just write it out. And what happens is we get the first term, this cost, has this variance in genetic variance. Then there's this covariance between what the the genes in individual I and the genes in its group, in its social partners, its group mates, then again times this transmission term, or sorry, plus the transmission term, yeah.
Sorry. The second term in the price equation is supposed to account for like non-random interaction, right?
Correct.
So why, maybe I misheard you, but what you're plugging in now is something that's supposed to account for the relatedness, and then you're plugging that into the first term, which is supposed to account for the natural selection term, right? Can you just walk me through that?
Yeah, so what will happen is, so this fitness term, which has an effect from social partners, some of whom may be related, this actually gets plugged into both of these pieces, right? Because both of them have a function of fitness. This one, wi here, wi here. What will happen is this term will be the one that has the relatedness coefficient, but this is for the chance that you're randomly interacting. Some of the individuals you randomly interact with will be relatives, some will not be. So if in the model you decide you actually have a preference for interacting with relatives, that there's a non-random interaction that way, then that would be accounted for in this second term. Yeah. So even if for example, if you don't disperse, if you're local and you don't migrate off after you become an adult and you just, have offspring with individuals you're nearby, by random chance, those individuals will more likely be your relatives than if you had moved. So it's that kind of spatial structure. So then the genetic relatedness here is really just this covariance between your genotype and your the mean group genotype divided by the genetic variance in the population. And if we do that, then we get from the price equation, we get this minus C plus RB plus this transmission term. So all we have to do to really recover the price equation is assume the transmission term isn't very large, so we would ignore changes due to mutation or other effects, and then we get Hamilton's rule straight away. And so the final one we'll talk about is group selection or multi-level selection, and it will sort of sound maybe a little rote at this point. That's hopefully true. So I wanted to get something from Price, because he's really the one who wrote down the equation. He's the one who also wrote down how you could partition it into between group and within group effects of selection. But he didn't write too many papers and died prematurely for all kinds of crazy reasons. So this is a quote from a paper from Bill Hamilton, who was using his approach in the 70s. So just saying that Darwin left an apparent difficulty unresolved. He saw that such traits for courage and self-sacrifice would naturally be counter-selected within a social group, whereas competition between the groups with most such qualities would be the ones best fitted to survive and increase. So the idea is you have a relatively simple kind of group structure. You have individuals within a group, and we'll just assume there's n of those individuals. And then there's n groups, and potentially there's movement or migration between the groups. So same kind of group structure model that Sergey was presenting earlier. And so we'll assume that there's some kind of, if we average across the fitness of all individuals within a group, like if this was group J, that average fitness is going to be WJ. And the average phenotype, so maybe the average level of cooperation in the group is going to be ZJ. And again, so for this purpose, we'll just assume straight away that transmission term is 0, just so we can keep the algebra a little bit simpler. So we'll start off just looking at this covariance term, the effect due to selection. And we know that there's, again, these little n groups of big N individuals. So that covariance term just writes out like that. It's the expected W times Z minus the expected W times the expected Z, just the definition of covariance. We add and subtract the same thing in that equation, and just read off what the new terms are. And the first one is a covariance between the mean group fitness and the mean group genotype or phenotype. And the second term is the expected covariance within groups so this would be the covariance within group J of the fitness of an individual in group J and its phenotype in group J. So what that leaves us with then is this first covariance is the evolutionary change due to between group selection, and the second term, this expected covariance, is the evolutionary change within group selection, due to within group selection. And so what's kind of nice about this, and some philosophers of biology have really kind of looked at this, is that this second term, you could think of, if you had not just two levels, if you had another third level within those groups, you could break this second term, this covariance term, into another set of covariance and expected covariance. So you might have three levels of selection happening, and so you could sort of imagine this as having, you could do this recursively for as many levels of population structure you think are important. So just sort of summing up, neglecting these transmission terms, that we've shown that those three fundamental quantity relationships, the fundamental theorem, inclusive fitness, group selection, we can all derive them relatively quickly using the price equation. So an important part is that it shows us how to partition, if we're looking at the effect of natural selection on the change in a trait. In this case, we'll be really thinking about cooperation. Then it shows us how to partition into these two components, a component due to selection and transmission. And then further, if we're thinking about population structure, we can either think about that structure, we can partition that change into some kind of more a spatial component where individuals are within groups, and that's what we care about. Or maybe we care about tracking how genetic relatedness or some kind of common ancestry changes the effect of selection, and so we could partition it that way. But either way, we're actually writing down the same thing on the left-hand side. We're calculating the same evolutionary change. Let me just take a drink here. Okay, so that's given us this framework then to have this multi-level model and to incorporate the first part that we talked about, this responsiveness, which would be due to individuals playing some kind of social game over and over again. So that, again, just a reminder that responsiveness was for a single population, so there wasn't any structure. So we'll include structure using the price equation. And we can use either this inclusive fitness partition we talked about with the Hamilton's rule, or we can use the group selection one. Either one will work. And we'll just use this inclusive fitness one since it's maybe a little simpler to start off with. So what we'll do is we'll write, remember the important thing was to have this individual fitness of individual J, and we're going to write that in terms of this responsiveness that it has when it's playing its social partner. So remember when we had initially, for a single population, when we had done these fitnesses, and we've just rescaled them, they had denominators before, we've rescaled them, we can sort of do that as long as everything is monotonic. Then these were the fitnesses of an intrinsic cooperator. This was the fitness of an intrinsic defector. So if we look, set this to be the fitness of individual I. So if it's a cooperator with if it has a 1 at this gene, so this has the cooperative gene, so then it has this fitness, and then the sum of these two, and then if it's not a cooperator, this is 0, and then it just has this fitness. If we just compare that to that regression expression we wrote for Hamilton's rule, then we can just match these straight away. So this was this GI would be PI here, so this thing has to be the minus C, and this was remember the frequency of cooperators in the population, so this is that mean, this gene frequency, the mean in the group, so this has to be the B. So then we just plug in the minus C and minus B into Hamilton's rule, and the invasion condition that we have then looks like this. So now it's more complicated than Hamilton's rule, and it has this responsiveness term in it. So what's kind of cool about this is that this expression is totally symmetric in the level of responsiveness and the genetic relatedness. So we can think of the effective population structure on creating conditions that favor cooperation having, given the way this model was simply set up, they have totally symmetric effects as to increasing responsiveness in that social game in terms of increasing the propensity for cooperation to evolve. And so additionally, you can't also, because of this multiplicative term, you couldn't just sort of replace the sum or some relatively simple function of sort of an assortativeness or some kind of correlation among types parameter. So these two different effects, at least at this level, are having an interaction between one another, so that they're having separate and interacting effects on cooperation. So equally, you could start with that group selection partition, this between group effect and within group effect of selection. Plug, so we'll suppose that the social groups are just are of size 2, so pairs are interacting. If we plug in our costs and benefits, our equation for this, the fitness of an individual I like we did before, then we get the between group selection component is this. It's the benefit minus the cost. That's effectively what like a group of cooperators might have, times 1 plus r. So this is going to increase if there's more relatedness. And then the within group component is b minus b plus c times 1 minus r. And so this is always negative. So given this kind of cooperative dilemma, this sort of prisoner's dilemma setup, there's always negative within group selection. That reflects the fact that cooperators are always doing more poorly within groups than they are doing between groups. So then all we have to do is plug in those minus c's and b's in terms of responsiveness. And what ends up with the between group selection is now we have the b minus c are the payoffs we had from each of the plays of the game. And this symmetric, just like before, that we in addition to relatedness, 1 plus r, we have 1 plus rho, the responsiveness in the game. So similarly, the within group selection component, we replace these fitness effects with these big B's and big C's, the costs and benefits from the playing the game once, and then again there's that 1 minus rho is coming out as well. And this whole thing again is still negative, so within group selection still being detrimental to the cooperators. So then you can figure out then the condition for when the between group selection outweighs within group selection, that's whenever you get an increase in cooperation, and that's just this expression here. And again, it simplifies entirely to the expression we had before. If you want to look at larger interaction groups, you can do that, and then the expression becomes a little bit more complicated if those interaction groups are a size n, but the whole process runs through exactly in the same way. So what's sort of nice about this is you can see then we talked in the very beginning, we talked about these transitions in individuality, looking at when conflicts within groups became minimal enough that the group itself might be an evolutionary unit in its own right. And so we can give kind of more of a mathematical sense for when that's happening by eliminating within group selection here. And the way we can eliminate it, now that we have both sort of genetic relatedness and this responsiveness from gameplay, is we can either set relatedness equal to 1 or responsiveness equal to 1. Either one of those is going to get rid of within group competition, within group selection. So you might think then that thinking about different kinds of organisms and their ability to cooperate or not, that relatedness might be more important of it reaching a value of 1 when we think about the evolution of multicellulars and the fact that we all go through single cell bottlenecks as multicellular organisms, and that single cell bottleneck affects a relatedness of 1. And then, but for multicellulars who have this more plastic behavior, that's maybe where if it were possible, maybe it's not, but if it were possible for them to their group to become an evolutionary individual in its own right, they might have another route to that, where they could have this be so responsive to one another that their evolutionary interests are completely aligned, and there's no within group selection. So this would allow either one of these routes would allow for the emergence of these groups as individuals, and for group level adaptation. And the idea is sort of to then think that at some level some of this must have been happening in some of these major transitions in evolution that Maynard Smith talked about. Okay, so this is sort of the second part. Does anyone need a break for a couple minutes, or we can keep going? I'll probably just roll through. Maybe it might be another, shouldn't be too long.
Sorry, can I ask a quick question?
Yeah.
So, is it possible at all to so I remember the diagram we had for the between group selection. There was
Here?
no specific structure that you could sort of pick, right? You're just assuming that you're basically just differentiating between within group and between group, and that's more or less it, right?
That's correct. Yes. I mean, that was the simplest group structure. You could have a more complicated group structure. There could be different types of groups, groups of different sizes, all kinds of heterogeneities, and then you would have to take that into account when you calculated. There might be different covariance terms then. They may have different weightings on them to account for the effect of groups of different size, or the effect, so you would just get a much more, but you would still have that same kind of idea of partition. And if that's interesting or important, then this is a tool to use. But if not, then maybe not. So. Okay, so now we're talking a little bit about the same idea, but in a more detailed model that allows us to study the dynamics. So this previous analysis with the price equation was totally static. We assumed there was some distribution of these genes, or there was some allele frequency to each individual at a certain time step, and the price equation allowed us to figure out what that distribution, what that mean of that distribution was the next time step, but only the mean. No other, we didn't know anything else about how the distribution changed. So that's just sort of saying what that said here. So it sort of works okay if we want to calculate potentially maybe an equilibrium in the mean at least. We could use that approach. We can potentially look for when that equilibrium could be unstable, but we can't calculate what that change in mean is over many successive generations without saying something more, without really having to start looking at these higher order moments of gene frequency. So there's no guarantee then of convergence. So even though we might be able to look at an equilibrium in the mean with that static approach, we don't know whether from a distance away from that equilibrium point whether the population will converge to it or not. So convergence then really requires this dynamic analysis. So we have to do this moment closure. We have to start looking at what the variance looks like, and we have to make an assumption about whether the variance is changing or not in order for the whole thing to be tractable. So this is where making an assumption about the strength of selection has become really important, and we're going to talk about how that allows us to sort of figure out how there are different time scales working in the population. So in quantitative genetics, they often, like in the breeder's equation, that assumption, this heritability h squared, the assumption was that heritability was constant, that the variance in the genetic variance of the population was staying the same. And implicit in an assumption for that is the fact that selection has to be weak. So weak in terms of its effect on that distribution changing over time. So if it's weak, then you can sort of assume that variance isn't changing very much and it's relatively constant. So the way that we're going to measure the sort of weakness of selection is through looking at coefficients that measure the effect of genotype on fitness, and we're going to assume those coefficients are small. And so those, for example, the selection coefficient that we'll use to measure the smallness of selection for that one locus model we had, that was this S. This was the selection coefficient. When we looked at Hamilton's rule, those C's and B's, these could be then considered as well selection coefficients. These are the things that are going to be assumed to be small. So the way that we can do that then is we can take some master parameter that sort of scales all those selection coefficients, and that's going to be sort of our one parameter that controls the overall strength of selection in the system. And we're going to write a Taylor series or asymptotic series in terms of this change in the mean as a function of that parameter centered at when that parameter is at 0. So then this first component is when there's no I'll probably write that here in a second. This first term is when there's neutral evolution. There's actually no selection since we've sent that strength of selection to 0. And then each subsequent term is sort of a correction on that. It's a perturbation. I guess I don't have that in the thing. Each term is a perturbation on that neutral effect. So that this is the sort of first order effect of selection on the change, the second order effect of selection on change, and so on. And so in this first term, when there's no selection, the only forces changing allele frequencies or genotype frequencies are things like mutation, migration, recombination, genetic drift. And so the nice thing about this kind of approximation is that this term is usually relatively easy to calculate. So, and other components, the change in the variance is relatively easy to calculate compared to the full model because there's no selection happening in it. So this is the study of a lot of work in population genetics, studying how these neutral models work. And in the simplest case, if we think about a single allele and there's no mutation, then that change under neutrality is just 0. So that's even sort of the simplest possible example. So typically, if we're going to cut off all the higher order terms, and we're going to say that selection is weak enough that all that matters is this neutral change plus some first order effect of selection. If we do that for the mean, then typically what happens, and this is sort of a result of a lot of work on different kinds of models, is that if we look at the change in the variance in the allele frequency, then there's some change in the variance due to neutral evolution, and there's a change in the variance due to selection, but that change in variance is a higher order. So this is a change that's order omega. This change tends to be of order omega squared. So it tends to be much smaller. So when you take this weak selection approximation, you get you sort of get these very slow changes in higher order moments due to natural selection, which this gives you the justification for assuming these things like genetic variance, these higher order moments, that they're constant. And this is sometimes called a separation of time scales, where there's a time scale for how the mean changes due to selection, and that's sort of relatively quick, and there's a time scale on which selection changes these higher order moments, and that's relatively slow. So then these higher order moments, because selection is changing them slowly when it's changing the mean relatively quickly, sometimes the phrase will be used quasi-equilibrium. In some of the old population genetics literature, it's called quasi-linkage equilibrium when looking at the effects of recombination between genes. And the recombination would much more quickly be the neutral component changing allele frequencies between genes so that they freely recombine, that selection can't affect their interactions if selection is weak enough. Whoops. Wrong button. So this is really the source of our moment closure, that this allows us to get rid of these higher order moments. We can assume these variances, these moments are constant. Anything that's a third moment, fourth moment, they all are 0 because their effect is so weak. And so it allows us to deal with a relatively simple set of equations to predict how the change in the mean works over many subsequent generations. So even though we assume selection is weak, there still might be an additional complexity that there could be multiple mutations in the population segregating. So there might be mutation A here, and then at some point, as it's increasing in frequency because it's beneficial, there's another mutation C and another mutation B. And this means that these mutations, if they were, like we had heard before, if there was a heterozygote advantage or if there was some other negative frequency dependent effect, they could form a stabilizing association. They could become what's called a stable polymorphism. Those two alleles might exist at the same, selection might stabilize their frequency at some intermediate value. So because this kind of complicates the analysis, we can make an additional assumption that as long as the population is small enough or if genetic drift is strong enough relative to mutation, so this drift is this force of stochastically removing variation from the population because individuals are randomly not getting sampled to become progenitors for the next generation. So drift always removes variation from the population. If mutation isn't adding variation quick enough, then when mutation is small enough, there will only be a single mutant in the population at any particular point in time. So one mutation here, and then if we wait long enough for this mutation to take over, to fix, then there will be some time where it's fixed before we get another mutation in the population. So this assumption of this sequential invasion and fixation of mutations, the mutation is weak enough that it's happening at that rate, is sometimes called a trait substitution sequence or sequential fixation. And this is the exact same assumption that you make in adaptive dynamics, which is the tool that Sergey was talking about earlier, that you're only allowing a single mutant individual to be in the population at any point in time. So there's actually some nice mathematical work that shows when this approximation holds, when this regime holds, and then it's just here that the total population size times the mutation rate times the log of the population size has to be much less than 1. So what you can just see here that as mutation rate gets much smaller, then you'll get much closer to reaching this, and as the population size grows large, it'll become more difficult. So you need drift to be strong, and so drift is strong when this population size is smaller, and mutation also has to be weak. So there's two kinds of go ahead.
That's when there's no balancing selection.
So if this assumption is true, there's only one mutant in the population, regardless of whether there's balancing selection or not. because statistically you'll never, even if there was heterozygote advantage, the allele, the second allele that you would need for an advantage, the first allele would fix, and then the second allele, because drift is strong enough, that allele will also fix over time. One of those alleles will be lost. Simply because it's a stochastic system, it can never maintain polymorphism once you run it long enough. One of those alleles, even with balancing selection, will be lost.
Yeah, so the question is what long enough is that?
This is the condition that ensures that it's long enough.
I was under the impression that was the condition after less than balancing selection, in which case it's fixation times an exponential, so it's much
I guess actually I don't know. I have to look at that. I'm not sure whether that they exclude. You're right. I don't know if they excluded those the balancing selection cases. I think they. Yeah, I guess most of sort of the approximate proofs of it show that it's for a mutation that has a unambiguous advantage. So you might need a stricter condition, but you still end up with another condition as well, right? You just have to make the drift strong enough in the population relative to selection. But I mean, as a reminder, that problem holds for adaptive dynamics as well, right? So any method. So given we're sort of assume that this is true, then even on this time scale that we're having mutants potentially invade, potentially fix or not, then if we're just looking at a fixed set of alleles, a fixed set of different possible mutants, then we can sort of think of this as a kind of short-term evolution where there's no possibility for sort of phenotypic novelty outside of that finite set of alleles. But on a longer-term time scale, we might imagine there's sort of a continuum, an infinity of possible different alleles, in which case this could be kind of a long-term approach, and there might be new phenotypic novelty. So what we'll focus on is this short-term approach here, where we're only going to look at these two different alleles, sort of big A and little a. They're two different variants. And we're going to assume that sequential fixation regime. So assume there's these two different types. This NT is the total population size. There's a mutation rate between the two alleles, A and little a, equal rate between the two alleles. And then, so again, you're having this process of mutation and then potentially fixation to switch the population to the alternative type. So as long as this approximation holds, you only need to track the frequency of time that the population is in either this state with all individuals of big A, or the fraction of time the population is in this state with all individuals of little a. And so the way that we can do this, we can set up a small transition matrix to track that, where the entries in the transition matrix are these fixation probabilities. So to go from a population fixed at big A to one at fixed at little a, would be the probability of big A, of a mutation arising of little a, and then probability that little a fixed. And so the mutations have actually been factored out here because we assume there's an assumption that mutations are very, very weak, very small, so that mu goes to 0. So the way then that you figure out whether you're at more big A here or little a is you look at then the stationary distribution of that matrix. So we calculate that out. It's just the relative ratio of these fixation probabilities. And so the long run frequency in this way of thinking about it of big A is just the expected frequency of it, which is just this first term here. So the probability that you go from little a to big A divided by the sum of those probabilities. So if we want to make an evolutionary success condition out of this, then we want to say, well, maybe we want to know that big A is going to be more common, it's more successful. So its frequency has to be greater than 1/2. And all that means is that the fixation probability of, so I think I wrote this the other way around. So it should be this, oh no, I wrote this the correct way. Of little a, of big A invading and going into, big A invading and going to a population of big A, starting from a population of little a, that has to be greater than when little a invades and takes over the population. So really determining which one is going to be more successful means calculating these two fixation probabilities. So then what we're going to want to do that is we're going to want to calculate these two fixation probabilities under the assumption of weak selection. And we're going to express these things in terms of quantities that we're used to measuring in population genetics. In particular, we're going to express them in terms of the time that two alleles take to go back in time to coalesce to a common ancestor. So the first step in this is writing this fixation probability in something that we can start to decompose into component pieces. So the first step is to say the fixation probability is really just the probability that in the long run, assuming that you have maybe a single initial mutant, that in the long run it's what the chance that if it fixes, then that frequency goes to 1. If it doesn't fix, that frequency goes to 0. So then that fixation probability should the average, if you averaged over many realizations of that process happening, it'd be the average frequency of this allele in the ultra long run as time goes to infinity. So then you can expand this. So this is basically the frequency at time infinity, and you can expand this as the initial time plus a sequence of changes of allele frequency over time, going all the way to t equals infinity. So this sum here. And then additionally, so this is, although we know there's an initial mutant, there's all kinds of stochastic changes happening here where the distribution could be one thing at time t, another it's changing at time t plus 1, and so on. So then we can decompose that. So then it's that change given the probability that it's in a certain state, the population is in a certain configuration at time t. So the nice thing is that this second piece here, this expected change, given the population is in a certain state, this is exactly the price equation that we talked about earlier. This initial frequency of the allele is really just the neutral fixation probability. So if there's no natural selection, the chance that a single copy of A will get fixed is its initial frequency, is 1 over the population size. So then this allows us to start working to write down this mutation fixation probability as this first order Taylor series. So it's this initial frequency, it's that neutral chance that it fixes, plus some first order effect of selection on the fixation probability. So after some work, we just differentiate this thing, and after some work and some couple additional assumptions, then what we end up getting is that it's this sum over time of these effects on the change in the mean allele frequency over time. So this is the change in the mean allele frequency. The price equation, so we'll use the second half of the price equation. So the price equation had, on the left-hand side, it had the change in the mean, and it assumed that change, that you knew what the population state was, that at time t you knew what the alleles, what frequency they had. So that's why we have to, it's only at this point it gets to be the price equation. Because otherwise before, here, this is conditioned on the initial frequency of the mutant at time 0, and then the change at time t. So this makes no assumption about what the state of the world is at time t. It has a distribution of states. So we have to, in order to actually get the price equation out of it, we have to extract that and sum across the probability of every single configuration the population could be in at time t. And then we just use the price equation. We replace this thing with the covariance. And then the next step is just to write fitness as this fitness of individual I as a function of its genotype and some selection coefficients. So for the moment, to make things simple, we'll only assume that individuals are interacting. If they're interacting socially, they're playing a game, they're only doing it in pairs. So what that means is that the fitness of this individual I can only be a function of the genotype of another individual times the selection coefficient. This would be if this individual was maybe a helper, then this might be frequency 1, and this might be a benefit. If this is the focal individual, this frequency would be whether it's a helper itself, and this coefficient would be the cost. And then additionally, if they're pairwise interactions, there could be multiplicative interactions. There could be effects of the fact that I'm a cooperator and my partner's a cooperator, and we get some additional benefit from that. So then we combine that fitness with the fixation probability and do some work and simplify. And what you end up getting, what's important here, is that there's a neutral component here, and then the effect of selection to first order is basically these weighted sums of selection coefficients times what are basically probabilities that individuals are identical genetically. So this is the expected, this is the chance that individual I and individual K1 both have the allele. Maybe if it's allele for cooperation, then it's that allele or some other allele. And so then this would be the probability that individual I, K1, oh, and that's a typo, that should be K2. So those three individuals all have that same allele. So that the additive interactions only depend on these pairs of genes, and the multiplicative interactions depends on triplets of genes. And what's nice about this is so far we haven't said anything about the population structure, because we're just summing across all individuals. So these things, this is a general way to write it, and you would calculate these things given whatever assumption you want to make about the structure. And so one issue with this so far is it's still this sum is still time dependent. So that's kind of gross. We want to get rid of that. And so the way that we're going to get rid of that is we're going to express those quantities as coalescence times. So what a coalescence time is first is just that's the chance, the time it takes for for genes and two individuals at the present to go back in time to the past to where they have a common ancestor. So if we think about the probability that two individuals at some time have the same gene, then given there was an initial, and this gene is the mutant, given that there was only one mutant in the population originally, the only way that the two individuals can have the same mutant gene is if that gene, the gene that you're looking at is the mutant originally, that you sampled the mutant way back in time, and that they coalesce before that point in time. So they both have to have the mutant gene, and then they both have to have coalesced back in time in order for these two things, for them to both have it. So the probability that it was originally the mutant is just the initial frequency that it was, if it started off as a single copy. And then this is the probability that we had coalescence before time t of those two gene lineages. So after rearranging and some other work, then these things all sum up to be expected coalescence times. So we still have these selection coefficients like we did before, the costs and the benefits, these multiplicative effects of the behavior on their fitness, and they're all now multiplied by the coalescence between individual I and K's gene, and between individual I and then K1 and K2's genes. And then we can get an analogous expression for the other fixation probability, and that allows us in sort of a general way to evaluate this condition of when is A going to be more common than little a in the long run? When is it going to fix in a population more commonly than the alternative fixes? So we can actually extract from this Hamilton's rule, and we'll see how to do that. So that canonical version of Hamilton's rule, remember, we used regression, linear regression, so to assume there were only additive effects of the social behavior of cooperation, not cooperation on fitness. And it looked at only these two different kinds of individuals, whether assumed individuals were sort of relatives or non-relatives. So to do that, we'll use the same group structure population we had before. There's n groups of size n. And if we assume that, and everything is symmetric other than that, then there's really three kinds of individuals in the population. There's the focal individual, this individual I here. There's individuals who are in the same group as the focal. And then there's individuals who are in some other group in the focal. And other groups are essentially equivalent, since that the mutation starts off as a single copy, and those other groups all have the other copy of the gene. So then the fitness of that individual is basically this weak selection measure. It's this cost, if it has the cooperator allele, the benefit times the frequency of the other individuals in its group who cooperate, and then this last term is the effect of essentially competition with other individuals in other groups who were sending migrants into that group. So we just plug this this fitness into the, whoops, that's sort of funny. We plug it into that condition for the fixation probabilities. And we get Hamilton's rule straight away. Where now Hamilton's rule, this relatedness, was actually expressed in terms of these coalescence times. It's expressed in terms of the time to coalescence between alleles in different groups, and then the time to coalescence between alleles in the same group. So if the population structure is very strong, which means maybe there's not very much migration between groups, individuals within groups are kind of going to be strong, highly related to one another, then the time that it takes alleles to coalesce between different groups is going to be very long, and the time that it takes alleles to coalesce within groups is going to be short. And what that does is that then makes up for a high genetic relatedness. And this also in population genetics takes another term. It's called in this simple kind of model, this island model, it's called FST. Okay, so just the final section, we're going to look at this, how this works with a little bit more specific example in terms of the actual costs and benefits from a social game. So what we really want to do is unpack these little c's and little b's like we did before when we looked at responsiveness. And in Hamilton's rule, and we need to do that, we need to specify how this demography works a bit more. We have to specify how mating and reproduction happens in more detail. So the first step is to specify the demography. So we're going to assume that adults are in groups, they interact socially, they cooperate or don't cooperate. The effects of that have effects on how many offspring they produce. And then those offspring, when they grow up to be juveniles, those juveniles migrate at rate m to another group, and at rate 1 minus m, they stay in their natal group. This kind of demography takes a name called hard selection. We'll talk about this in a second. And then finally, once juveniles arrive in their new group, or if they stayed in their natal group, they're going to compete for the n different, basically, patches that are available in that group to be adults in the next generation. So there's going to be a complete turnover of the population. So the generations are non-overlapping. Then for the social interaction that affects how many juveniles you make, we'll assume there's these pairwise interactions between adults. Individuals with this allele big A cooperate, and there's a little a defect. And so then the fertility is their average payoff from that interaction of however many, on that average payoff they get from however many cooperators and defectors there are in their group. And that payoff is going to be, if there's two cooperators, we're going to modify it a little bit. We're going to not make it non-additive. So before we had this just B minus C, minus B and B, we're going to add a synergistic effect here. So the possibility is that two cooperators can maybe get even more by working together than just the additive effect of the helping that they had before. So what that means is that in normal cases where this synergistic effect isn't very large, we still end up with a prisoner's dilemma. But if the synergistic effect is really strong, if it's greater than the cost, then we end up with something called a stag hunt or coordination game, where there's actually the Nash equilibrium from the game change, and there's a Nash equilibrium for individuals cooperating in the one-shot game, and a Nash equilibrium for individuals defecting. And as well, you could actually change the costs entirely. You could change them in sign, make them negative costs. Sorry, sign should be flipped, I think, again. But you can flip, by just changing the value of D and changing the sign of the cost, you can flip the whole game to something called a snowdrift game or a hawk dove game. So you get sort of all possible game setups basically by just altering the payoffs in here. So given that, we can write then what the fertility of this individual is. It's the benefit times the frequency of individuals who cooperate in their group minus the cost times whether they're a cooperator or not, plus the synergy if they cooperate and however many group members are in their group cooperate. So then finally, we have to calculate these coalescence times we showed before. There's some methods for how you do that. Turns out that these are the expressions for those coalescence times. This is basically the time for these within a population. This is sort of a classic result. The time it takes for those to coalesce is the total size of the population. And then if there's two individuals in two different populations, the time to coalescence is this, where this M you can think of as just kind of measuring the effective number of migrants from a group. So if M is big, then there's migration is strong and there's weak group structure, and if M is small, then migration is weak and individuals are sort of tightly held within groups. So finally, to put it all together, we put all the stuff back into those fixation probability equations. We drop out a bunch of these terms to simplify it, and we get these two different fixation probabilities. So relatively simple. These are that neutral change, and this is the effect of selection. Yep. Because you take the series expansion around omega equals 0. Yes. I don't know if there's other methods to do it. It's almost impossible to take it. If you did it on any other value, you'd be always evaluating terms around some selection, some positive value of selection. And since that's intrinsically what we're trying to do, we don't know how to solve the full system when selection is acting, then we take the expansion around 0, and then, which is basically neutrality, and then just add essentially corrections onto that. And so that's why it only works for weak selection, is because those corrections have to be small, sort of relatively small. Strong selection, it's not going to work, potentially. So a couple observations. There's no benefit term in here at all. So the benefits of cooperating have been exactly canceled out by basically the cost of competing with kin. When you cooperate within a group, that benefit increases how many offspring you produce, but some of those offspring stay within your group, and you compete with them for the spots in the next generation. So this is a classic result going back to some work from Peter Taylor in the early 90s. This cancellation, in this kind of population, you get this exact cancellation of benefits, of the benefit of helping. So you can also recover this 1/3 law. I think you guys talked about this earlier. 1/3 law from Novak. If you that was taken in a single population, the way you get a single population here is you allow migration to become very strong. So it becomes there's no structure anymore. And when you get that, then the condition that Novak had was that the fixation of this cooperator type had to be greater than neutrality, and that's just this D greater than 3 over C, or than 3C. If you translate that back into their payoffs, that's exactly the 1/3 law. As well, when you actually look at the full condition for success, and this is really the important condition, not this one, then the condition is that the synergy has to be greater than twice the cost. And if you translate this back into sort of more classical evolutionary and otherwise game theory, this is called risk dominance. When people started looking at, there became a big problem of we know we can solve games, and games have potentially multiple Nash equilibria. How do we know which Nash equilibrium is the one that sort of rational agents should choose? So it's called the equilibrium selection problem. And one way that people thought of is that I think you guys talked about the replicator equation. If you take the replicator equation and you look at the basin of attraction for cooperate, big A, and defect, little a, then one way of choosing which equilibrium, if there are multiple, you should choose, is you look at the one that has the bigger basin of attraction under the replicator dynamic. That, in this model, this ensures that big A has the bigger basin of attraction, D greater than 2C. So what this shows is that even though we've had this island model population structure, we get a result that is as if we never added population structure in the first place. So this is a very general, so this is a generalization in some sense of this Taylor result, that this structure isn't adding anything to the population, this specific kind of population structure. And it's really due to this exact canceling of the benefits or any change from population structure with the benefits as on the ultimate success of the individuals. So it's true that while it's true for this particular population structure, it's not true for other population structures. So that those assumptions we made in the demography, those matter crucially. So two assumptions that you can change immediately that will get rid of this. One is if we make it so that we have overlapping generations, so adults will live at the same time their offspring do, that things no longer cancel. The other thing that we're going to look at is changing this hard selection. If this is this idea that juveniles they're produced after their parents have a chance to cooperate, and then they either migrate or don't migrate. So alternatives to this, one simple one is that first, after juveniles are produced, there's density regulation. So what that means is that after they're made, they all compete for spots, those n spots in each group, and then they migrate. Now, once that competition has taken place, each group will contribute exactly the same number of migrants because it's been regulated down. Each group's been regulated down to exactly n individuals in the group. So that means that cooperative groups that might have more juveniles, they get regulated down to n. Defector groups that might have had slightly fewer juveniles, they also get regulated down to n. So effectively, it means that it should be harder for cooperation to work. Then the other alternative is that maybe there's some kind of competition among groups for resources, like Sergey was talking about. And then there is additionally within group competition for whatever the group obtains. And so that actually should help cooperation. So we'll see that come out with these two different examples. So for soft selection, where we had that effect where individual groups, no group is more productive despite its level of cooperation than any other, that condition becomes this, where this was the risk dominance one, the effect of essentially no population structure plus this. So it makes it harder for cooperation to evolve, so that D has to be even larger. And effectively, this is increased competition within the group. So this would in some sense even incentivize harming within the group. So cooperation is worse, the conditions. So if you allow for this group competition, though, then the condition turns out to be this. So remember, this is, if just the D greater than 2C was the unstructured result, now you've added on the left-hand side this effect of what two cooperators get working together, divided by the number of migrants. So if population is strong, then this migration M, the number of migrants, is small. And as you make it smaller and smaller, you can effectively make cooperation, you can increase the chance that cooperation is predominant arbitrarily large as long as the migration rate becomes small enough. So this cooperation becomes much easier to evolve. So we could rearrange this in terms of what this relatedness this is equal to in this particular population. It's 1 over 1 plus 2m. And you get an expression that looks like this. So the cost, it looks kind of like Hamilton's rule. There's the cost, the benefit times the relatedness, plus this synergy coefficient times 1 plus relatedness over 2. In general, you can actually, as a general result, you can always write it this way in this kind of model with just pairwise interactions, where there's some kind of coefficient times the benefit plus the synergy coefficient times 1 plus that over 2. And this coefficient now we call kind of scaled relatedness. So in a sense, it measures both what relatedness measured earlier, which is the effect, it measures some kind of effect of common ancestry, but it also takes into account these effects of local competition and other effects of demography on fitness, and the fact that those things might actually cancel. So this kappa, this is equal to, it sort of maps 1-to-1 with other things like the structure coefficient from Karina Tarnita's work. And so we can actually look at specific examples. So this kappa, it's 0 in that hard selection case. That was just the D has to be greater than 2C. It's a negative when there's soft selection, when cooperation is harder. And it's equal to FST, or just relatedness, when there's group competition, which is when cooperation becomes easier. So again, that's just sort of reemphasizing that this is a general result whenever you have this model assuming sequential fixation. And the nice thing about it is that it sort of nicely summarizes evolutionary success in a relatively complex model, because there's stochasticity, there's finite population size, there's mutation between two different types. Those types are playing a social game within groups. And what it does is it partitions the effect of selection into the payoffs from those games those individuals play, and it encapsulates all of the effects of demography, all of the effects of population structure into a single index, which is this kappa. So to finally tie up the whole thing, if we look back at that result we had when we just looked at the price equation without any sense for how populations were changing over many generations, when we had the responsiveness component part of it, this was our increase condition. So what we know now is that this relatedness, if we want to take into account the fact that this competition locally can make it harder for cooperation to evolve, we have to replace this relatedness coefficient with kappa. And this ends up being the condition we have to satisfy. And then moreover, if you look at some of this other kinds of population structures we've talked about before, we've talked about star-shaped populations, evolutionary graph theory, all of those are cases where we have irregular graphs when there's the connectivity between populations isn't symmetric. Then that means that, for example, this population here receives more migrants than these populations on the edge. And that creates systemic changes in the change in whether or not you get a big A or a little a just due to migration, just due to population structure. So those things can all be incorporated, but it makes the whole thing just quite a bit more complicated. And then finally, I'll leave in the slide, there's stuff for further reading in case you're interested in hearing more about this. And then I'll just leave it at that and take questions. Yeah.
Silly question. And that the type of plot that you put up, and I'm trying to figure out what these types of plots are called.
Oh.
With the sequential fixation.
Yeah, I don't know. I don't know if they have a name.
What are they called?
Oh, Muller-Fisher. Yeah, so they're in. Well, it's okay, but is it? So I got it from a Crow and Kimura paper. Was it in a paper before that? It was. Okay. So this actual plot is from a Crow and Kimura paper. I don't know what the original. It must be a Muller or something paper that originally had this. Yeah. Yeah.
So, about coalescence, can you elaborate a bit about what is your goal there? You say something about calculating how long is it before it is, I mean,
Oh, yeah.
how far is the origin, but that sounds like not really the subject here, right?
Yeah. So it's I guess what I would say is when we start this, the whole goal here was to calculate an expression for fixation probability, and essentially it turns out that in calculating that expression, you end up with expected coalescence times, and there are ways to sort of convert those back into things like probabilities of identity by descent, which maybe are a little bit easier to get a handle on. But I actually don't know of a way to, in this general case, when you have these multiplicative interactions, here, a general case of actually getting those probabilities of identity by descent. So the simplest thing is just to leave it as coalescence times. It's a bit more computationally complicated just to deal directly with those. Yes.
So, yeah, I'm confused. What is the difference between coalescence and
Well, a coalescence time would be literally like the number of generations it takes going back in time for two lineages to coalesce. The probability of identity by descent would be the probability that they're identical by descent. So that would, the coalescence time doesn't, they have a relatively simple, I'm trying to remember, they have a relatively simple relationship. It's like, but because there's one of them, there's an assumption that when you look at the probabilities of identity by descent, that only equals coalescence time when the mutation rate goes to 0. So there's a limit on mutation, and it's sort of handling that expression that becomes difficult when you put it in here to try to get it in terms of identities by descent. It's a little, in this case, it's more general, yes, because the identity by descent, the reason you care about identity by descent is because you're assuming that there's no mutations along the way. So in this case for coalescence, you don't actually have to assume that it's already built in because you know there was initial mutant, and if they're both A, they can't have mutated along the way, and they both have to be A.